Thank you for the kind introduction. Good morning, everyone. Today, in my presentation, I'm going to talk about developing and implementing science-based standards in bioequivalence assessment. I will be covering three topics in my presentation. First, we will talk about the role of Abbreviated New Drug Application (NDA) assessors in product-specific guidance development. Next, we will cover the impact of PSG recommendations on NDA assessments along with case studies. Then we will transition into case studies related to alternate BE approach proposals to PSG recommendations proposed by the generic drug applicants. Finally, I will summarize my presentation. So let's start with role of NDA assessors in PSG development. In addition to a group of dedicated scientists and researchers, the assessors who assess ANDA submissions are also involved in the development of new PSGs and revision of existing PSGs. Prior to the publication of PSGs in public domain, the NDA assessors ensure that the PSG recommendations are aligned with the current NDA assessment practices. In addition, the NDA assessors evaluate the impact of PSG recommendations on pending as well as approved ANDAs. In next couple of slides, we will discuss two case studies that outline whether the change in PSG recommendations, that revision of PSG, had an impact on NDA assessment. In the first case study, the drug product is an extended-release oral capsule formulation. The posted PSG recommended single-dose two-way crossover fasting and fed BE studies in healthy subjects. Over the period to fulfill the post-marketing commitment, the RLD, New Drug Application applicant, conducted a BE study comparing administration of the drug product as whole intact capsules versus as granules sprinkled on soft food, that apple sauce, in this case in healthy subjects under fasting conditions. The results of this study demonstrated bioequivalence on using two methods of administration, which supported the labeling revision to recommend administration of granules with 1 tablespoon of apple sauce as an alternative route of administration for patients who are unable to swallow an intact capsule. During the NDA assessment, NDA assessor evaluated this new information in the RLD labeling and proposed to revise the PSG to add fasting sprinkle in applesauce BE study. At the time of PSG revision proposal, there were several approved and pending NDAs. None of the NDA submission had data from BE study conducted under fasting sprinkle in applesauce conditions. Nevertheless, the NDA assessor's recommendation to add the fasting sprinkle in apple sauce study is in alignment with scientific recommendations for modified release drug products and agency's BE assessment practice for similar cases. Therefore, the PSG was revised to recommend fasting sprinkle in applesauce BE study in addition to fasting and fed BE studies. In this case, the applicants for pending as well as approved NDAs were requested to submit the results of fasting sprinkled in applesauce BE study and update their labeling to include this new administration method that sprinkle on applesauce for their generic drug products. In this case study 2, we will see that revision of PSG had no impact on the pending NDAs and there were no approved NDAs for these drug products. Let's say this is a drug product X, which is a novel oral anticoagulant. I will refer to it as NOC during the talk. Per posted PSG for this drug product, two-way crossover fasting and fed BE studies are recommended in healthy subjects. Whereas the PSGs for other NOCs recommend four-way rather than two-way crossover in vivo BE studies. The four-way fully replicated crossover design for other NOACs is recommended due to steep exposure-response relationships for both efficacy and safety. Use of reference-scaled average BE approach to widen the BE limit is not recommended in these cases. Applicants should use the average BE approach with BE limits of 80 to 125% and upper limit of the 90% CI for the test to reference ratio of the within subject variability should be less than or equal to 2.5. We at the agency do revisit published PSGs to ensure that recommendations do align with the latest current thinking as new information becomes available, which was also mentioned in the earlier presentations. In this case also, the PSG was revisited to determine whether PSG revision is needed to be consistent with the recommendation for other NOACs. NDA assessor analyzed all the data available to the agency and determined that drug product X has different physicochemical, safety efficacy profile, and inter-subject variability than other NOACs. Overall, drug product X has less safety concerns and risks are not high enough to recommend additional BE data, even though it belonged to the same drug class as other NOACs. NDA assessor then coordinated with relevant offices in CDER to reach an agreement that no revision of PSG is needed for drug product X in terms of study design. This avoided the need for multiple applicants conducting new in vivo BE studies without compromising BE standards. This is the type of outcome I think we all like if it does not compromise our bioequivalence standards. The PSG in this case was also revised not to change the study design, but to add recommendations regarding additional clinical lab tests for inclusion criteria to ensure subject safety without changing the study design. That the same two-way crossover study design was kept as it was initially recommended in the PSG. As there was no change in the study design, the change in PSG recommendations had no impact on pending NDAs. Now we will go through 4 case studies where generic drug applicants proposed alternative BE approach than that recommended in the PSG and agency's evaluation and decision regarding proposed approach. In the case study 3, which is the case study 1 under alternate BE approach category, the drug product is a topical cream. The PSG for this topical cream was posted a couple of years ago, recommended comparative clinical endpoint study to demonstrate bioequivalence based on the agency's scientific thinking and available data at the time of PSG posting. As we know, clinical endpoint B studies are more expensive, time-consuming, and often burdensome than in vivo BE studies with PK endpoints and in vitro BE approaches. With the evolvement of use of in vitro methods in bioequivalence assessment for the topical drug products, a generic drug applicant through pre-ANDA meeting request proposed an alternative in vitro BE approach. Agency evaluated the submitted information and deemed the applicant's proposal acceptable. Subsequently, the applicant submitted its NDA with proposed in vitro BE approach. As I mentioned earlier in the presentation, the agency revisited the already published PSGs as new information becomes available that would require PSG revision to align with the current thinking. Accordingly, in this case also, the PSG was updated to recommend in vitro BE approach. In this case study for alternate BE approach, the proposed study population is different than that recommended in the PSG. The drug product is an anti-cancer drug. When the agency first published the PSG, there was very limited data on exposure of this drug in healthy subjects that indicated high adverse events. This raised serious safety concerns in healthy subjects, making it necessary to recommend BE study in patients who are already receiving the drug product as part of their therapy per the RLD labeling. Recently, years after the publication of the PSG, a generic drug applicant submitted a bio-IND proposing BE study in healthy subjects. The bio-IND submission included data from multiple BE studies conducted internationally in healthy subjects. The agency evaluated this newly submitted data and determined that it is safe to conduct BE studies in healthy subjects at the lowest feasible clinical dose and deemed the applicant's proposal to conduct BE study in healthy subjects acceptable. Subsequently, the agency revised PSG for this drug product to recommend BE studies in healthy subjects. We all understand that it is quite challenging to conduct BE studies in cancer patients, as in this case, and clinical endpoint BE study as in previous case. So, we at agency do appreciate applicants coming up with scientifically supported alternative BE approaches that also satisfies the requirement of the applicable statutes and regulations, which helps agency update the PSG recommendations, and all this can help reduce the cost and time of generic drug development and increase availability of high quality affordable generics for American public. This case study is related to alternate BE study design for multiple drug products. The PSGs for these drug products recommended two-way crossover design for bioequivalence studies. During the conduct of pilot BE studies, the applicants observed high variability in pharmacokinetic parameters. Therefore, through controlled correspondence pathway, these applicants requested the agency's guidance on conducting partial three-way or fully replicate four-way crossover designs rather than two-way crossover design recommended in the PSG. In some cases, for fixed-dose combination drug products, for BE evaluation, proposal was to use data from only first two periods for analyte 1 and from all periods for analyte 2 that showed high variability. The applicants were communicated that it is acceptable to use partial or fully replicate design, but a reference scale BE analysis approach should only be applied to a specific pharmacokinetic parameter that exhibit high intra-subject variability that equal to or greater than 30% for the reference product in the pivotal BE study. Please note that it is not necessary for the applicants to submit controlled correspondence for this type of questions, as use of partial or fully replicate study designs are usually acceptable alternatives for all PSGs that recommend two-way crossover studies. However, the applicants should consider if there are any safety concerns with additional dosing periods, as it can be concern for some drug products such as anti-cancer drugs. This was not the case for the drug products for which the applicants requested agency's guidance. In the last case study, we will learn about alternate BE approach for lower strength. This is an extended release drug product for which BE studies are recommended on the highest strength, and the lower two strengths may be deemed bioequivalent to the corresponding strengths of the reference product based on acceptable BE studies on highest strength, acceptable dissolution and formulation proportionality across all strengths. The applicant submitted a controlled correspondence to seek agency's concurrence on their proposed approach to demonstrate BE for lower strengths. Per the applicant's proposed approach, all strengths, let's say for example, these strengths are 10 mg, 15 mg, and 30 mg, will not be developed proportionally similar but will have the same inactive ingredient. Therefore, a dose proportionality study was proposed for lower strengths to meet requirements of the PSG. Other data will be submitted as per PSG. The proposed study design for dose proportionality study was three-way single-dose crossover in healthy subjects under fasting conditions, where each subject will receive different strengths of the test product but at the same dose. That 30 mg given as 3 units of 10 mg, 2 units of 15 mg, and 1 unit of 30 mg in a crossway manner. The agency determined that RLD applicant also demonstrated dose proportionality among different strengths using similar approach. The applicant's proposal to submit the NDA using the proposed approach to demonstrate BE for the two lower strengths was deemed reasonable. To summarize my presentation, PSGs provide scientific recommendations to help industry develop generic drug products efficiently. PSGs describe the agency's current thinking on a specific drug product, and serve as guidelines for NDA assessors in the evaluation of BE data. NDA assessors apply a science-based approach in the assessment of BE study. As we learn through case studies, the applicants may use an alternative BE approach if it is scientifically justified and satisfies the requirements of the applicable statutes and regulations. The applicants can discuss their alternative BE approach through appropriate pathways such as pre-NM meetings, controlled correspondences, journal correspondences, etc. In my last slide, I would like to thank Dr. Honglin and Dr. Bing for their contributions to the presentation. Thank you very much for your attention. I will be back at the end of the webinar to respond to any questions you might submit. Thank you.